Maybe I'll wait a minute.
Somebody else? Oh, coming, they're coming.
They're coming. Nice. And they are cookies. Okay, well, maybe I'll get started and introduce our seminar speaker today. We have the pleasure and honor of having Dr. Emilio Grande from CSU East Bay today, from the Department of Earth and Environmental Sciences.
What?
But I forgot, now there's a new title.
Oh, they're changing to Earth, Environmental, Sustainability Sciences.
There you go. Dr. Grande did his bachelor's and master's in geology at CSU East Bay, followed by his PhD in earth sciences at UC Santa Cruz. So he's going to tell us about some of his research at CSU East Bay now. So thank you so much for being here.
Yeah, thank you, and thank you for inviting me. Also, I apologize that my slide has a little animation, so hopefully no one got seasick. Yeah, it's not supposed to be up there for that long, so I apologize. Okay, yeah, so before I start talking about the presentation, I just wanted to say that the research that I'm presenting here today, most of it anyway, was carried out in a place that we call Elkhorn Slough, but I just wanted to acknowledge there were people that lived in this area before we showed up, and I just wanted to say that we pay our respect to the native people of this land that have occupied this territory for immemorial times. Okay, so with that being said, today I'm going to talk about most of the work that I've been doing for approximately the past 5 years since I started my PhD, and I like to call it learning about coastal hydrology from high-frequency measurements. And I would tell my friends that I like, my joke is reading between the tides. That's my little quirky things. So yeah, as you can see from here, I work in coastal environments. And today I'm going to be talking about coastal hydrology and a little bit about biochemical processes that happen in coastal places. First of all, I wanted to acknowledge the questions of why do we care, right? It turns out that these coastal systems are really, really important, and there is many reasons why they're very important. Students, especially your classes of geology and environmental sciences, you have learned probably about most of these. Coastal environments, they're really critical for flood protection, erosion control. They are really important habitat for different, especially migratory birds. They are also really important for fisheries. I read in statistics once that something like 50% of all of the fisheries in the southeastern United States have something to do with salt marshes, which is actually really good. They're also really important for water quality. It has been found that salt marshes can remove excess pollutants and improve water quality. They are also really important for recreation. Who doesn't like doing some boating or some kayaking? And also really, really important for carbon sequestration. And even though I'm really personally interested in both the water quality aspect and the carbon sequestration, today I'm going to be focusing in some of the water quality aspect or benefit of salt marshes. Okay, so a little bit of more motivation for this is actually related to climate change. One of the things that, and hopefully I can say climate change in here and be safe. Yeah, climate change is projected to increase hypoxia in coastal environments. And of course, hypoxia is low levels of oxygen that can result in images like the one that we're looking at in here. And this is a fish. I once had someone ask me if that was a piece of chicken, and it's not. It's a dead fish. It does look like chicken, but anyways. All right, so this is actually related to the fact that excess nutrients inputs into coastal environments have caused environmental concerns. The image that I'm showing you here is my field site, and I'm going to introduce that in a second. And what we see here in this green mat is actually covering the mud flat of the salt marsh. And this is during one of the now somewhat common algae bloom events that we see in my field site down in Elkhorn Slough, unfortunately. As I mentioned earlier, salt marshes have been recognized to be hotspots for nutrient processing. This is really critical because not only the scientific community has become aware of this, but also the general public. These are some screenshots that I have taken from the news over the past few years, just highlighting issues with algae bloom events because they started affecting our communities. I have some images in here from some news from where I live, like the East Bay and the San Francisco Bay Area. And this thing is all over the place. A few years ago, we had this really bad algae bloom event in Lake Mary, which is this lake in right downtown Auckland. All the fish died, and I was for once very surprised that there were fish living in the lake. And then that actually it smelled really bad, and everybody became aware because it's in the middle of the city. So anyways, it's something that the public is becoming very aware. So the thing is that even though we know that these places are so important, we still have a limited understanding about how spatial temporally variable many of these critical processes are. Spatial temporally is a big word, just how variable in space and in time the things are. Just in case you feel intimidated by that word, don't worry about that. Now, luckily, we live in a time where advances in technology have made possible that we can measure solutes at really high frequency. I personally have some probes that can measure nitrate concentration in water every few seconds. So that's, at least to me, that's high frequency. My colleague that measures fluxes, earlier we were talking about eddy covariance flux hours, they measure several times per second. So to me, a few seconds is high enough. But anyways, the thing, however, is that these high frequency measurements over space, and just bringing back to the salt marsh, let's say at different salt marsh elevations, so different positions within the salt marsh, those are limited. And it's not because scientists are not interested in that. It's mostly, this probe that I was telling you and bragging about, it's very expensive. I only have one. So I cannot deploy those probes everywhere, right? It's also field intensive. In my lab once, we wanted to do a 24-hour sampling campaign and then just sample every 1 hour during a tidal cycle. And that involved getting volunteers, undergrad volunteers, all of the PhD students in our lab. We came up together, and at the end, the results didn't work well because the lab wasn't working well, and pretty much it was a disaster. So it takes a lot of work to do that type of analysis. So I was lucky enough that for the past few years, I worked with a few people that are very smart, and they allow me to use their instrumentation, and we developed a setup that helped us measure solute concentrations at really high frequency in groundwater from different marsh elevations within this salt marsh that I've been talking about. So this setup, and don't worry, I'm going to show it to you in a second. I'm building up to it. This was, we have this field site in Elkhorn Slough that is located in Central California, right in the center of the Monterey Bay between Santa Cruz and Monterey, that's Elkhorn Slough. And the field site itself is in the upper parts of Elkhorn Slough where the black star is up there. If you've never been to Elkhorn Slough, it's one of these places that I do recommend people visiting. It is really beautiful. You can actually rent kayaks up and down in Moss Landing towards the mouth of Elkhorn Slough, and then you can kayak in, and you can see sea otters. It's really beautiful, and you see the baby sea otters. They're really, really cute animals. Anyways, the kayaking is not too bad. If you go too far up, and then coming back down, sometimes it gets windy and it can be tricky. But if you stick around the mouth, you can have a really fun time. I'd really recommend it. You can have that on your list. All right, so Elkhorn Slough, well, at least parts of Elkhorn Slough, are associated with the National Estuarine Research Reserve System, and this is an organization that, to be honest, at this moment I really don't know where their status is with the current administration and all, but they used to be managed by the state and also by NOAA, and then they have a system, I think, close to 40 different of these reserves around the country, and Elkhorn Slough is one of these. As a result of this, there has been ongoing monitoring of water quality within Elkhorn Slough for, I think it's something between 3 and 4 decades of monthly measurements of water quality. In Elkhorn Slough's water quality is a concern because Elkhorn Slough is surrounded by agricultural development. So runoff, especially during the winter, has been found to bring a lot of excess of these nutrients that result in the algae blooms and so on. So we do have a record, we have an idea of what's going on in surface water within the estuary, okay? They put out these cards with qualitative data for the general public so that everybody within the community understands the efforts that are happening within Elkhorn Slough. So there are a few things that I just wanted to mention because that will help me develop my study site and the design. So one of the things that you should know about Elkhorn Slough is that the concentration of ammonium tend to be high in surface water, and that is true everywhere in surface water. Additionally, nitrate tends to be elevated, especially in the mouth of Elkhorn Slough. That's where the main river that drains these agricultural areas discharges into Elkhorn Slough. And then what has been found is that the concentration of nitrate decreases as you move inland. Nitrate gets processed, let's call it for now, okay? And then the concentration of nitrate alone decreases drastically ammonium does stay high everywhere within Elkhorn Slope. Okay, so let's zoom in into my specific field site, which is just a little piece of a salt marsh. And in here, I just label different what we call marsh positions in the community. Simply, those are different marsh elevations. So they have like this. There is this micro topography that results that a low marsh, which is what I call lower, it's inundated a little bit more frequently or for a longer time than an upper marsh position. And I'm talking about here about only a few centimeters of differences, but that is enough that in some places that turns out to be important for the biochemical processes that are happening, because some places are inundated less frequently. In addition to that, I have also labeled other components of the salt marsh, including the mud flat, the transition zone, like right the transition between the terrestrial and marine environment, and then just regular terrestrial, big trees and so on and so forth. Okay, not shown in here, a little bit to the left of this, then there is like a really big farm. They used to do strawberries, and now they're doing Brussels sprouts. So it's very common around Elkhorn Slope. But basically, I have instrumentation, and I'm going to show you some of the instruments at the lower marsh position, the middle marsh position, the upper marsh position, and also in the upland. And in there is it's fresh water. So it's dry at the interface, but we still have fresh groundwater in there discharging onto the marsh. Okay, so this is what this is a profile view of that transect. The x-axis is not to scale. And so what I have at each marsh position, I have a few other probes out there besides this, but for the purpose of this talk, I wanted to show you that at each marsh position, I saw observation groundwater wells. These were deployed at down to 70 centimeters of depth. I also have these redox potential probes, and they are represented by these gray bars. They have those rings, and they measure redox potential on those rings at 10, 30, and 50 centimeters of depth. This measured redox potential continuously. I was measuring redox potential every 1 minute. So again, for my standards, high frequency. Inside the observation wells, I also measure groundwater levels continuously and temperature. And also, in addition to that, I had this system of what I call sampling cups. They're somewhat like a sampling piezometer, but a little bit different. This is what a sampling cup looks like. I built this myself, and I just copied them from some colleague that used them before. I used a PVC, and my idea was to try to create a closed chamber. So inside that this PVC over here, I glue a PVC sheet, sort of like what is shown in here, and I added a ton of epoxy to seal it off, and I create like a closed chamber. Okay. Then I drilled through that seal, and I put two pieces of tubing. One went all the way to the bottom of the cup, and I used that one to pump water outside. And then the other one that will have a smaller diameter, as shown in here, was there just to allow some air to flow out as the sampling cup will fill up with water, so that I won't create a vacuum and I didn't have to apply pressure. Then I seal around those pieces of tubing. I glue another piece of PVC to give it some structure. I seal it up again at the top, and then I just deploy them in the field, under the surface that were installed at 10, 30, and 50 centimeters, same depth at which I measure redox potential. And in the image, you can see some of the tubing going up to my central platform. You can see a solar panel up there. And then on that platform, I have a few other things. So those sampling cups, again, were connected with some tubing down to that platform. So in that platform, I had this a manifold pump that had a system of valves, actually 12 valves, with a little Arduino computer that kept all of the valves closed and will open only one at a time. So I just wrote a little program, and the pump will trigger and measure every 4.5 minutes, and only from one valve. And then the sample will run through a field-based spectrophotometer, which is this silver probe that uses UV visible to measure a few things, including nitrate and dissolved organic carbon. And then the pump will reverse, send the sample back through waste and just dump it on the salt marsh, and then pump from another sampling cup soon after. So I had a total of 12 valves. A few of them, one was to sample air because I want to correct for the signal of air. Another one was a cleaning solution, so on and so forth. So I will sample the same groundwater cup in less than an hour. It will come back to the same. Okay. So yeah, it is a really neat database. At least I'm not aware of that, of actually any other groundwater continuous monitoring at this level. So at the time I was very excited. I have to come clean, and I'm going to show you this. When I first started working on this, I had this idea of a whole year of groundwater so on. This stuff, it is a pain. It's a reality of this. You know, the salt marsh has fine sediments. Things clogged, and I had to replace them. The pump, you can imagine, it's a very small peristaltic pump. It was pumping all the time. I went through like 4 of them during my PhD. I've gone through 2 of them now that have students working on this. The tubing I had to replace every 2 weeks. So anyways, the best that I did was I managed to have approximately 10 days of continuous deployments every other month. Okay, and I'm going to show you the frequency of that. And at the very least, it gave me some continuous measurement snapshots over the year. Okay, and I'm going to try to show you what I did with that. Okay, so this is enough, you know, speaking and explaining the methodology. So let's start looking at some more fun things, and let's move on to some results. So the first thing that I wanted to show you is very briefly. I wanted to talk about the redox potential data, because you know, in the salt marsh I mentioned, it's a very harsh environment. Many things break. It's one of the biggest challenges maintaining your instrumentation. It's really hard. That's saline water all the time. It is a pain. However, these redox potential probes, they work like a champ. Like you know, the only problem that I had was I had to replace the batteries. Other than that, they continue to work. I was very surprised. So I'm really happy with my redox potential measurements. So I just wanted to show you a few things in here. We have 3 panels in here, and this is just a time series of like a year or so of redox potential. The light blue line represents redox potential at 10 centimeters, dark blue at 30 centimeters, and the purple at 50 centimeters, and we have a panel for the different marsh elevations. And there are a few things that I wanted to go mention in here. First of all, just for our reference, here is a simplified redox ladder, just to have an idea of what's going on. So first of all, I wanted to say that, not surprisingly, redox potential really represent anoxic condition, like very low values of redox potential. What I mean by this is if you look at, for example, the regular values of redox potential under which oxygen is found, okay, see that it's way above 0, okay, so notice that in here we're really relatively below 0. Only on the shallowest depth we do see some variations and some periods where we do see some higher values of redox potential. That's the first thing that I wanted to mention. This is, of course, not surprising. Like most wetlands, this is what you will expect. Very low levels of oxygen, very anoxic. You smell that rotten egg smell, right? Okay, and also the other thing that I wanted to mention is that overall there is a very small variability in redox potential. And this really didn't surprise many people, because most biochemists that have been working in salt marshes that I talked to will tell me that, yeah, that's what I would expect. Like, nothing happens in the salt marsh. Sediment is so fine, it's almost like a clay. There is no groundwater movement in there. And I mean, I didn't listen. I wanted to understand that better. Still, we do see that in the shallowest depth, again, it's only 10 centimeters, we do see these big swings. But the thing that really puzzled me and that I pursue was this very small scale variability that I saw on most of these time series. Over short time scales, I saw those little wiggles, and I wanted to see if those were real or if it was just an artifact. So I went down that path, and to explain that, the first thing that I did was look at things in the frequency domain. And this might get a little bit intimidating, but trust me, it's very simple, and I'm going to explain everything. I have to say that I'm not a mathematician by any means. In this day and age, like a Fourier transform, you can just do it in MATLAB. It's FFT, and it's really easy. Anybody can do it. So, but anyways, the first thing that I did is I applied Fourier transform to all of these high-frequency measurements that I had. Okay, Fourier transform again is very simple. You get a time series or a signal, and you can just decompose it into a series of sine waves, which is what this image is showing. And then you can just simply identify the frequencies of variation of those different sine waves. Okay, and then you can just create a plot and look at what are the most important frequencies, and that might be able to tell you something about what is happening. Okay, so let me show you what the figure looks like, and again, it's very straightforward. We have in here a panel for each of the different depth and for the different marsh positions. In the y-axis, I have power spectral density. Just think of that as power. The higher the power, the better the thing. Let's say it that way for now. Okay. Again, we're no mathematicians. Hopefully there is not a real mathematician here that might get upset. Okay, we're all in the natural science world. Okay, we just apply their stuff. And then in the x-axis, we have a period in hours. Okay, so then I notice that in each plot, I have these vertical dashed lines. Those correspond to 12 and 24 hours. Okay, so what I wanted to mention in here is that I noticed that this is the ghost one that I have. At those 12 and 24 hours, we had these peaks. Okay. in the redox potential. Okay, and I saw that across all of the depth and marsh elevations. So that sort of like got this bug in my head of, well, maybe there is some variability actually that is happening at these frequencies. And the one thing that I know that happened at that time could be the tides. It's a tidal system. The site gets flooded and then drains. So I kept thinking, is this telling me something about a connectivity between the hydrology and the biochemistry? Perhaps there is something in there. Now, the limitation of the Fourier transform, and this is well known, is the fact that there is no time information here. What I mean by that is, for example, let's take this panel in here. We know that those frequencies were important, and they should receive high spectral power at those frequencies, but we don't know if that was maybe like 2 days out of the year, and that is enough to generate this figure. Or maybe it's something that happened every other day, or half a year and not the other year, right? There is no time information in here, okay? So I'm going to show you one other, a little bit more complicated figure, but that will be it. After that, everything will be very straightforward. So the work around that is other fancy math that can do also transformations in the frequency domain, and that's just wavelet analysis. So Fourier decomposes the signal into sine waves. Wavelet analysis, you decompose your signal into wavelets. And wavelets, I mean, we don't learn about wavelets as we do with sine waves, because in trigonometry, you know, we learn a lot about sine and cosine. Nevertheless, I don't have to do any of this by hand myself. You know, there are computer programs that you can write and do that stuff for you very simply. So again, now what I'm going to do is get my signal, same signal, and just use this wavelet to decompose the signal, and then gain some information. Now this figure looks a little bit different, but again, it's very simple, and I'm going to explain it to you. What we have in here, this very trippy, it looks like tie-dye, really like that. It looks really nice. Now, what we have in the y-axis is periods in hours. In the x-axis, we have time. Okay, that's good so far. And then I'm using colors to represent power. Okay, so the higher the color in this scale, the higher the power. And actually, again, I didn't develop any of this. Do keep that in mind. I'm just using stuff that are, you know, somewhat out there for everybody to use. You can actually detect things that are statistically significant. And actually, in here, I'm putting inside this black polygon, so anything that is inside that black polygon is the stuff that is statistically significant. Okay? All right, and the figure on the right is what we call the global wavelet. And for the purpose of the talk, we can assume that this is representing like a weighted average of what we're seeing on the left. Okay? All right. So there are a few things in here that are really interesting, and I want to draw your attention to. Okay. In addition to that, I also want to mention, I forgot, this red line is also marking statistical significance. So stuff to the right of that line is what is significant, stuff to the left is not. Okay? And what we're seeing here is that in the global wavelet, we again have those peaks, 12 and 24 hours that we showed you when we did the Fourier analysis. That's really cool. The other really cool thing is that we also see that in the continuous wavelet transform, as we will expect. The really nice thing about the wavelet transform is that now we have time in here. These are timestamps, okay, from my time series. And what we see is that those are not constant through time. There are periods for which we don't see that variability happening, namely that time up there. And as it turns out, that particular time corresponds to, you know, the end of the fall, beginning of our winter periods, when in this part of the world we actually start having our precipitation, for example. So what we start seeing is that not only there seems to be this connectivity between the tidal forcings, let's say the tides. Sorry that I'm using the word forcing, I mean to use more jargon. So we have the tides probably influencing the biochemistry, but it also seems that there is some seasonality, or yeah, let's call it seasonality for now, that can also be putting some, might also be affecting the biochemistry, let's put it that way. Because for some part of the year, we don't see the redox potential, and if we think of redox potential as a representation of biochemical processes that could be occurring in the subsurface, we see that difference, okay? There is one more figure that I wanted to show you related to redox potential, and I wanted to zoom into one precipitation event. I have to say that, okay, I don't have years in here, but this is all from 2020, 2021, very dry period. This is the time where I was doing my PhD. Like in our lab, we were chasing storms, very few storms to chase. Now what, not only was this during COVID, but also there wasn't many storms to chase, and there wasn't really that much water to be studied. Anyways, luckily in the salt marsh we have the ocean, so there's always some water. Okay, back to the story. Here we have again redox potential. This I don't have a label in here, but this is for the upper marsh position. Not that it matters. In the y-axis again we have redox potential in millivolts. Sorry that I changed the colors. This is just from a different paper where I use different colors. But basically we have in green the shallow redox potential, in purple the 30 centimeters, and then in blue the 50 centimeter depth. And what we see in here is that before the rain event, that is represented by the blue bars on top, and I have that shaded region, we see that strong tidal signal on the time series. And then as we move through the period when it's raining, we see that behavior changing. And actually the amplitude of that, let's call it sine-like function, gets completely dampened. One thing that maybe you wonder is, well, maybe it was raining, but maybe there were no tides. I actually have, of course, I measured water level in there, so I want to show you that indeed the tides continue to occur during those during that period. In here I have in pink the marsh groundwater level, and of course when it floods, my transducer also measures that the water level is above the surface. That's why this is the surface of the marsh, and you see that the water level is above the surface of the marsh. And also we see that the terrestrial groundwater level increases a little bit as a result of that precipitation event. So the tides continue to occur. Whatever impact the tides may have in the redox potential got dampened during the precipitation event. Now, there are a few things that could happen in here. I do not know what actually happened because I wasn't really there. But there are a few things we can definitely walk away from this figure. One is that either when it rains, rain on the salt marsh infiltrates into the subsurface and somehow changes the chemistry of the soil. Of course, that cannot be completely true because you can just rain some amount, but then you're mixing the rain with the whole ocean during a high tide. Okay? But nevertheless, it could be some dilution in there, and something might be happening on the chemistry of the infiltrating surface water. Or it could be that the terrestrial groundwater level is increasing, and as a result, there is some lateral pressure onto the salt marsh. And I'll come back to that idea later because that pushed me into other projects that I'm going to show at the end. Nevertheless, in either case, what I walked away from this analysis is the fact that we used to conceptualize salt marshes as these places where very little happened. Most people, even now that I talk to, tells me, oh yeah, just sample maybe the first 5 centimeters, below 5 centimeters, there is not much variability because water is really stagnant in there. What I found with this study is that is not completely true. Perhaps that is true in some places, at least in the field site where I worked, and some others around the country that I have looked at afterwards, and I'm going to come back to that at the end of my talk. That doesn't seem to be the case. Okay, so for now, some takeaway number 1, let's call it. Redox potential is coherent with groundwater. Coherence is a word that they use for the wavelet analysis things. Let's say that they vary together, okay? And number 2, redox potential can be affected, just plain English word for now, by these episodic precipitation events. Okay, so let's come back to the hydrology, which is my bread and butter. Here, we have a time series of groundwater level. The dark blue is the terrestrial groundwater level. The yellow is the upper marsh. The light blue is the middle marsh. And the pink is the low marsh. And then I have these horizontal dashed lines. Hopefully you can see them. Those are marking the elevation of the salt marsh. Okay, just wanted to reference the elevation of the salt marsh. And then on top I have a precipitation hyetograph, and this extends for a year. It's actually a little bit more than a water year. Well, actually less. Well, December 20 to December 2021. That's water year in there somewhere. There are a few things that I wanted to mention in here, and some of this will be very evident for most of you because you live in California and you know that in the winter it rains. And what we see is that when it rains, the terrestrial groundwater level is elevated. And then we go through these periods, like after the spring and into the summer, where it doesn't really rain, and then the groundwater level also decreases, mostly because that also coincides with the period when plants are evapotranspiring. So this shallow groundwater level is it's supposed to decrease. So no surprises in there. Actually, in this place, that change is at 1.3 meters, which is it's not small beans. It's something. Okay. The really interesting thing in here is when we start looking at what is happening in the salt marsh. So what we see is that during this wet season, when the terrestrial water level is elevated, we see high tides, flow the transect and then they drain. but the elevation of the water, and I'm using here that black line in the upper marsh to show that the water level within the marsh stays at a base level corresponding to the elevation of the salt marsh. And once we transition to the dry periods, when the terrestrial groundwater level is lower, we see that, of course, there are high tides flood the transect, but then during low tides, we see that the salt marsh actually drains below its elevation. And really talking about some connectivity that probably exists between the terrestrial groundwater and the hydrology of the salt marsh itself. Okay, and I haven't forgotten, I have all that instrumentation that I started just saying that, the really cool, it's coming up here. So my deployment, in here I have these gray bars just representing the different deployment periods that I have. I did manage to capture a few of the wet periods and a few of the dry periods, so at the very least we'll get some idea of what might be happening in the salt marsh. Okay, so here is some of the results corresponding to that. Here we have a time series for a few days of nitrate, represented by the blue dots. This is from the upper marsh at 10 centimeters. And I also have here a water level time series from the marsh itself. And then on top I have a hyetograph of just a precipitation time series as inverted bars. And very briefly, what I wanted to mention here is that we do see that the nitrate time series sort of follows some tidal pattern. It seems like after a high tide or a high water level, with a little bit of a lag, there is a decrease in nitrate. And then as we transition into the low tide, the concentration of nitrate sort of like starts increasing, it goes back up to some level. And then that pattern sort of repeats itself. And then what we see is that once we move to the period of that precipitation event, that relationship gets a little bit disturbed. Okay? And I have to say that I saw very similar patterns across all the different depths and also across all the different marsh elevations. Okay? Again, this very close relationship between these solutes, okay, and the water level. So here's another figure just showing this relationship. And then in here, as I have said many times, I am a hydrologist, and there is some metrics that we have. Hydrologists have been looking at relationship of water level and solutes for a very long time, and that relationship is okay. So sorry, I went ahead of myself. Yeah, we see close relationship between the tides and the nitrate concentration. And as I was saying, in hydrology, we have taken advantage of these relationships to gain information. The relationship between water level and solutes is in most cases hysteretic, it's usually non linear, and there are a lot of indices that we have developed that allow us to learn a little bit about what might be happening. Okay? Yeah, as I mentioned, there are these hysteresis indices that we have developed as a community that can help us understand what might be happening to the solutes during a flood event. So, of course, these indices were developed for riverine environments. You look at storm hydrograph, and then you look at some solute, and then you learn something from that. What I found in here, and what I tried to do, was to use that same idea to the salt marsh subsurface. So then I decided to use these tidal events as my storm hydrographs and look at what the relationship was between the water level and the continuous nitrate concentration for all of those periods. So the analysis itself is very simple. I'm just going to walk you through how this works. You will see it's very straightforward. So let's assume for a moment that we're measuring water level in the Little Chico Creek. That's the name of the creek, right? Yeah. So luckily there is a creek that runs through here. So let's assume that we are hydrologists on campus and we're measuring water level in the creek. And last night, for example, it rained, and maybe this is what the water level is, right? It was like low, and then it went up during the storm, and now it's receding, and maybe now it's sort of like back at base level. So let's assume that we're using some of these probes that can continuously measure the concentration of one solute, and we have solute 1, and maybe this is what solute 1 looked like over that storm event. It increased with the storm, and then it decreased, okay? And let's assume that we're measuring a second solute, and this solute behaves very, very differently. The concentration of the solute decreased during that period. Okay, these two, it happens all the time. If you have, I don't know, Sandrine will correct me here. I also like something like phosphate probably behaves like this, and maybe nitrate will behave like that, a little bit different. I have to say that they are not perfect, especially the blue one. I made this by hands before. I had even worse ones this morning, and I just really quickly tried to code this to have something to show you. But okay, so let's go further in here. We can now just make a plot of for solid want, the concentration of the solid versus water level, and that will look something like this. And again, as I said, it's not completely linear. It actually has some sort of loop. It's really, and you have all type of shapes, and it can get very interesting. And then solid 2, which again is not the nicest, it will look something like this. Okay. Now this actually is showing a lot of really useful information. One thing that we could do is just feed a slope through there, and it will look like this. That's not the exact slope of this. I drew those in PowerPoint. But so the slope of these are known as concentration discharge, also known as CQ plots, but it doesn't really matter. The slope of the plots tells you if there is a flushing pattern or a dilution pattern. If you have a positive slope, it means that there is flushing, right? The concentration of a solid is increasing with increasing water level. And if you have a negative slope, it means that there is dilution, right? The concentration of the solid decreases with an increase in the water level. Okay, so that is one metric. Another thing that we can look at is also sort of like the direction of the loop. That can also give you some information. So you normally get clockwise loops when the concentration of the solid is higher in the rising limb than in the falling limb, and then you get counterclockwise loops when the concentration is higher in the falling limb than in the rising limb. Okay, so what does that mean for the salt marsh? And this is the sort of like what I came up with. This is actually the relationship of one of actual tidal events. And here I'm just using colors, like a color scale to represent sort of like the directionality. Okay, so again, we use the hysteresis index. Before I jump into that, you do notice that I normalize both the water level and the nitrate concentration. That way, all the indices will vary between -1 and +1, and you will see. And then I just use them straightforward that way, and I can compare them. Again, I do not claim credit for that. It's sort of like what the community has been doing for decades now. Okay, just wanted to mention that. So, okay, directionality of the loop. In the case of the salt marsh, what we sort of like hypothesize is that clockwise loop will mean that the tides bring in some nitrogen, and then some of that gets removed over the tidal cycle. And then we assume that a counterclockwise loop will indicate that the nitrate concentration increase over the tidal cycle. And then there are a few reasons for why that will happen. Either it could be like, this idea of groundwater actually delivering some nitrate, it's like another source. Or it could be that nitrate is formed. Hopefully you have seen one of those nitrogen cycle things, and nitrate is all over the place. You have one form of nitrogen over here, and then another one right next to it. It's all over the place. So it could also be formed within the salt marsh relatively quickly. Okay, and I can actually derive that index really easily by looking at the difference in the solid concentration in the rising limb minus the falling limb, and then I calculate the average of that, which gives me one overall directionality. Okay, and then the other index will be the flushing index, and it's related to fitting a slope through the plot, and it will tell us if the nitrate concentration increase or decrease overall. Remember that a positive slope will mean that the salt marsh will be a nitrate source because the concentration increase over the tidal event, and a negative one will be that the salt marsh is overall a nitrate sink. I have to say that this is a very simplistic approach. I am aware that many other processes influence the nitrogen cycle, okay? But overall, this is something that I could learn just by looking at these plots, okay? All right, so then the really good thing about this analysis is that I can then just plot those indices in a plot like this, and then basically, depending on where they plot, I can, pretty much say something about some of the processes that might be dominating, okay? So yeah, here it is. This is the data. I use different points to represent different marsh positions, and then the colors represent seasonality. I did not find any significant, at least statistical, differences between marsh positions, or like marsh elevation, or depth. All of those were, again, statistically similar for the whole data set. However, I did find that seasonality was statistically significant, or significantly different, I should say. Okay, so what we found is that in dry periods, represented here by the color red, we have mostly clockwise hysteresis and then depletion patterns, suggesting that the soil marsh is probably a nitrate sink during those periods. And then in wet periods, we see that counterclockwise loops dominate and also producing patterns, suggesting that the soil marsh could be a nitrate source. So I'm almost done, by the way, don't worry. We can look at this into a conceptual model, just to, I spend a lot of time making the drawing, so I like to show it. And just in plain English, what we see is that in dry periods, we see that the soil marsh probably is really good at removing nitrate from surface water. But that is unlike what we see during the wet season when we believe that the soil marsh could be, it is producing some nitrate, definitely. At least at the different depths at which I measure, it's flushing some nitrate. I really don't know if that nitrate will go back into the marsh or maybe it gets consumed just right down gradient from what I'm measuring. But at least at those depths where I looked at, that's sort of like the general pattern that we observe. Okay, so I'm going to try to summarize everything that I've been talking about today. So we have this site, we have this transect, and I show you that it gets flooded with the tides. So those tides will bring in, well, for once it's surface water, so it has some dissolved oxygen in it, which is actually really important for the biochemistry. And it also has some nutrients, mostly by nutrients here I'm referring to the nitrogen species that it brings, particularly ammonium that I mentioned at the very beginning. Hopefully you remember that is high. And then, based on my observations, there is a fast vertical exchange of this surface water with the marsh subsurface. And that sort of results in biochemical reactions, not necessarily during the low tide, this is just an animation. Okay, so then the main points are that nitrate concentrations in port water seem to be controlled by the tides. We saw that close relationship with the tidal cycles. The tidal signals were observed also in the redox potential record, although not year-round. It seems like the relationship also changes with the seasons to some extent. Also, we saw that precipitation water seems to, again, disturb that general relationship that we see with redox potential and also with nitrate. And lastly, we see that seasonality tends to drive the biochemical behavior of the soil marsh. And by this I mean that there could be a shift between the soil marsh being like mostly just or exclusively removing nitrate versus maybe removing less or at least maybe perhaps producing nitrate. Okay, so this is what I have so far. In addition to that, some of this stuff that I learned sort of like pushed me into, okay, let me apply all the stuff that I spent so much time learning about to other soil marshes. Well, I see similar patterns. So that's what I wanted to show you about. The first thing that I did, I just reached out to my colleague, Julia Guimond. She did some work in Delaware, in the East Coast, and she collected similar redox potential data. So then I did this statistical analysis called mutual information. Again, it sounds more complicated than it is. It's just a comparison between time series, and it tells you how much information two time series share. Okay? But what I have in here is results of this so-called mutual information between other different time series and redox potential for my field site in Alcorn Slough on the top, and then St. John's. This is the St. John's River. It's another National Historic Research Reserve in Delaware. And what we have in here, this TGWL means the reservoir groundwater level, and MGWL, I apologize for this, means marsh groundwater level. The thing that I wanted to show in here is that after doing this analysis and some other analysis, what we found that the terrestrial groundwater level actually seems to share more information, and it's not only causistic, we did some other analysis actually looking at influences, and the terrestrial groundwater level seems to influence more the redox potential than even the marsh groundwater level. That, and even though we see that tidal pulse, we see that tidal signal, that seasonality controlling those variations in terrestrial groundwater level seem to matter. Okay, and I have to say that we extended this analysis to not only those two sites. I did the analysis for 7 different coastal marshes, mostly in the East Coast, and the only one in the West Coast is the one in Alcorn Slough. And we saw that in all of these places, in some places more than in others, of course, the geology is probably really important in other parts, but we see that this somewhat new idea that the terrestrial groundwater level in the salt marsh also plays a role. Surprisingly, people that study fresh wetlands, it's something that everybody's aware, because if the wetland gets disconnected from the groundwater level, then it's not a wetland anymore. So that has always been present, and what we have noticed is that in the coastal hydrology community, we have been focused in the oceans as dictating everything that happens, and we believe that is, while the ocean and the tides are really important, what is happening on the continent also seems to matter. Now, I do want to say that we don't believe that is that necessarily groundwater is the limiting solutes, and that's what's resulting in these changes in biochemistry. We believe that a more simple approach can be taken, and simple by differences in pressure. When you have an elevated terrestrial groundwater level, there is a higher pressure towards the ocean, so that exchange of tidal water with the subsurface is much dampened during the wet season than during the dry season, and perhaps that is what's driving this. So we actually published this paper earlier, well, mid-February in Nature Water, and we have a few proposals that we submitted to dig deeper into this, because we think that there could be some really cool things yet to be discovered. So we're really excited for this. So anyways, I'm gonna chart up in 2 slides. So the main takeaways of what I've been saying in here is that we showed that over short time periods, tidal forcing seemed to be a fundamental control of the biochemical processes and also the subsurface hydrology of the marsh. And lastly, that seasonality also drives changes in biochemical behavior in the salt marsh subsurface. So at the very end, this is just no research. I just wanted to share what I'm up to these days. I don't know if it's something like the other seminars do, but when I was in Santa Cruz, we had also weekly seminars, and that's what people used to do, so I figured I'll do the same. So yeah, I continue to do this work. I have the same setup and instrumentation, and I'm now trying to focus more in the carbon cycling. So I have a field site with a colleague that she looks at eddy covariance flux towers. We have a probe in a tidal creek, so we're measuring mostly dissolving organic carbon, but also organic carbon in and out of the salt marsh, and then I'm looking at what's happening with the groundwater. So we hope that we can also look further into what processes might be influencing the carbon cycle within the subsurface that sometimes it could be overlooked. In addition to that, most of the work that I actually do is not related to coastal hydrology. I do isotopes hydrology. So I have been doing this project. I actually published a paper at the end of last year, sort of like tracking the origin of different storms into the Bay Area, and then using the signature, the isotopic signature of the different storms as natural tracers to track the movement of water through different watershed systems, in this case urban watersheds. And lastly, I also work with other isotopes like tritium and noble gases to look at recharge elevations and also groundwater ages. And I have a project far from here in Mount Shasta and near Lawson, where I'm looking at spring water, and I'm dating, not taking out any date, but calculating the age of the spring water in the McCloud River and the Upper Sacramento River as well. So yeah, so that's how I stay busy. And yeah, so with that being said, I want to acknowledge. You can imagine, all of this involves a lot of field work. So I didn't do any of this alone. I had help from many people. Yeah, and this is the guy that invented the pump that I use. So he deserves a lot of the credit because he's the smart one in here. So that's it. I don't have anything else to say. Thank you so much.